So our last and fourth class of biomolecules are lipids. And lipids, by and large, are hydrophobic. So they are scared of water. But the number one rule in biology is that there's always an exception to the rule. So we'll talk about some guy that's a weirdo. All right, so we're going to talk about triglycerides. We're going to look at the structure of those. We alluded to this earlier, but how fats can be used as energy storage molecules. And then we are going to introduce this exception, which is phospholipids, that are really important for the production of cellular membranes, whether that's the plasma membrane on the outside of the cell or membranes on the insides of the cell. Phospholipids are so dang important. Okay, so we need to spend enough time to get to know those guys. Lipids are mostly hydrophobic. Okay, they are composed mostly of these CH bonds, which we know are nonpolar. Okay, so I'll just draw out the structure of a fatty acid real quick. And remember, anytime there's a bend, there's a carbon there. I'm not going to go through and draw all the C's. Yeah, maybe I will. Yeah, maybe. Why not? And we know that carbons can make 4 bonds. So this guy has 1, 2, 3, 4. This one has 1, 2. So we need to give him hydrogens, couple hydrogens. Okay, and so forth. Blah, 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 blah. I'm going to write them all. But this guy has 3 in the other. Okay, well, we can see this is totally composed of all of these non-polar bonds. And that's therefore that's going to make it hydrophilic. Animals use these as long-term energy storage. You'll also see seeds have a high amount of lipids. So your seeds and nuts are going to have higher lipids, and that's for energy for the new baby plant before it becomes photosynthetic. Okay, so I already drew a fatty acid for you, but fatty acids are these long chains of hydrocarbons. They have a, here's our carboxyl group. Oh, we're saying it again. Okay, here's our carboxyl group. There it is. Okay, we saw it. Remember, we saw those on amino acids also, and sometimes in carbohydrates. Okay, we're usually anywhere from 14 to 24 carbons long. As we'll talk about later in the semester, you can use these as long-term energy storage because these carbon to carbon bonds are very stable, but we can actually pull off 2 of these carbons at the end and then put that into part of the cellular respiration cycle that I won't scare you about anymore right now. Okay, so we need to talk about saturated fatty acids. Okay, in a saturated fatty acids, all the carbon-carbon bonds are all single bonds. Which means that these are holding as many hydrogens as they can. You can't change the chemical structure of this to add any more hydrogen atoms to this chemical structure. It's all filled up. It's saturated. Oof, I can't do it anymore. Okay. But in our unsaturated fatty acids, we have at least, so we have 1 or more double bonds between some carbons. Okay, so theoretically, if we broke that carbon bond, we could throw in a couple more hydrogens on here. Okay, it's unsaturated. We could fit more hydrogens on that unsaturated fatty acid. Okay. Our saturated fatty acids, when we have a whole bunch of them, because they have this pretty linear structure, that means that they're going to pack together nicely. They're going to pack tightly. That means that they are going to be solid at room temperature, because there's not a lot of space in between those individual molecules. Okay, so we tend to think of these colloquially as fats. So these are going to be your butter, your margarine, those kinds of things. Okay, and if we eat too much of these, then what's going to happen is that this big pad of butter is going to get stuck in our arteries and block our arteries, and resulting in a heart attack or a stroke, and that's probably to be avoided. Okay. Now, if we look at the chemical structure of our unsaturated fatty acid, we see that where that double bond is, we get this like really weird shape. And so these don't really necessarily pack together very tightly. And the more double bonds, the more crooked it is. Okay, so this bent chain is going to prevent tight packing. That means that we're going to be liquid at room temperature. These are going to be things that we kind of know commonly as oils. So our olive oil, canola oil, avocado oil, all those things that you might have in your kitchen that are going to be a little bit healthier for you because they're not going to pack so tightly. Okay. All right. So this is our first type of lipid that we're going to learn. These are called triglycerides, and there's a few components that we need to talk about here. So we have our fatty acid chains. Okay, these are remember, these are our carbons. They're all hooked together, mostly 14 to 20 carbohydrates long. They could be saturated. They might be unsaturated. Okay. It depends on the type of triglyceride that we're making. Okay, so those are our three fatty acids. We also have this glycerol molecule that has 3 carbons. And then we have these lovely little oxygens here. We can do dehydration reactions, boop out those 3 water molecules. So here they are, they've been kicked out. And now we have our fatty acid chains hooked to that glycerol backbone. So sometimes you'll see them drawn simplified, something like this. And that is important that we have tri-3-glyceride named after our glycerol molecule. Okay. Triglycerides, that's something that you're going to be monitored for, especially as you get older, because the concentration of those can lead to health issues. We need them, but we don't need too many of them. Okay. All right. Well, let's talk about phospholipids, because these guys, remember, these are really important lipids. These are so important that we need to make sure that we understand the structure of phospholipids. Okay. We are going to start with our same glycerol backbone. Except I can't spell. This is the same that we saw in fatty acids, or sorry, in triglycerides. So let's just throw up a triglyceride real quick here. Here's our simplified glycerol, and we had our 3 fatty acid chains. Remember, these little bends are supposed to, there's a carbon here, but I'm not going to draw them in. Okay. So check, we got our glycerol backbone. We have two fatty acid tails. Okay, and these, remember, these can be either, if we look at here, boop, they can be saturated, or, sorry. It's gonna be unsaturated, like this one. This one is saturated because we're all single bonds, all the way down. Okay. Now, on our glycerol backbone, we still have a spot left. Okay, so we hooked a fatty acid to this carbon, we hooked a fatty acid to this carbon. On this carbon, we are going to add a phosphate group. And as we might recall, our phosphate groups are a phosphorus atom hooked to four oxygen. In order for the octet rule to be satisfied, two of our oxygen atoms have negative charges. Haha, okay. All right, interesting. So our fatty acid chains, we know how they feel about water with all of these non-polar bonds. So they are going to be hydrophobic. I'm so scared of water. So scary. Okay, but if we come up here to our phosphate group, we see that we don't have just one charge, but we have two charges. So when we have a charged particle, it can be positive or negative, we're going to be hydrophilic. Yay, we love it. We love water. So if we have a, get rid of that. If we have a phospholipid in water, because these are negative charges, our partial positive charges of our water molecules are going to be coming and hanging around these negatively charged oxygens as part of that phosphate group. But the fatty acids are going to want to be hiding from water because they're scared. Therefore, we say that phospholipids are amphiphilic because they are both hydrophilic and hydrophobic. Okay, so an amphibian has two lives because it lives in the water and on land. So an amphiphilic molecule has both hydrophilic and hydrophobic portions of the same molecule. Now, this is really interesting because what happens when we take a whole bunch of these phospholipids and we chuck them in water, they're going to do something really awesome. They are going to spontaneously, without any extra energy input, and within milliseconds, they're going to form one of these three structures. But we're going to spend most of our time talking about this phospholipid bilayer. Okay, so these heads are those phosphate groups. So let me do this. And then this part is the fatty acid tails. So the phosphate heads are going to be facing where the water is on the outside and in here on the inside. Whereas the fatty acids are going to be interacting with each other and hiding from the water so that they don't have to interact with that water. Okay. Now, all of a sudden, this is the basic structure of a cell, where we have watery stuff on the outside We have some watery stuff on the inside. And all we have to do is trap some other stuff on the inside to be able to maintain homeostasis and form life.
That's so cool.